All right, for lecture 19, we are going to go over a real-world example of how you might use command line tools and the ability to call that in Python. So we are going to cover PCR, and the program is designed to create primers, to design and create primers that you might need in order to do PCR. So PCR is short for polymerase chain reaction, and this is a technology in molecular biology used to amplify pieces of DNA across several orders of magnitude. So you start out with, what might be a region of the DNA that has a very small number of copies. So you might have a genomic sample, a DNA sample that you take from a crime scene. You might have a DNA sample that you take from a patient. You might have a DNA sample that you take from a bone fragment, and you might be interested in studying just a small piece of that DNA. So, your genome is 2 gigabases of DNA, and you might be interested in a small region. So why might you be interested in a small region? So an example of this is Huntington's disease. So there's a small region of the genome that can be duplicated, and if you have a large number of duplications of this particular small repeat, then that means that you have a very high likelihood of getting Huntington's in older age. And so you might want to be able to amplify that particular region to see how long it is. And so PCR was designed that allows you to do that, and it requires the generation and the creation and the synthesis of 2 DNA primers. And these primers are complementary to part of the DNA, and they require their synthesis. So this also required techniques in order to synthesize small oligos that are about 20 base pairs long. And so these days you just order those primers and you specify their DNA sequence, and then companies will send back a tube that contains quadrillions of copies of these primers. So it's really easy these days to order small oligos that serve as the primer. And so you're designing 2 of these regions, and these 2 regions indicate the start and the stop of the region that you want to amplify. So you start with a piece of DNA. So this is a sample. So again, you take this either from, if you're at a crime scene, you might collect some sort of skin cells or hair cells, and these hair cells have genomic DNA within them, and so you isolate that genomic DNA. And so the primer has the same sequence as part of the strand, and that allows it to bind to that region of the strand. And so what you do is you create a hybrid that contains one of the original strands along with the primer with this red, which originally is only the size of this red region. And then you combine this with a polymerase, and this polymerase allows you to amplify. You make a copy from the opposite strand, and so you're able to copy this part of the DNA for a certain number of base pairs before it falls off. And the same thing is true with that reversed primer. So this is a primer that is complementary to the top strand, and so it forms a double helix, and then the polymerase is able to extend from the 3' side in order to make a copy of this. So let's say you just start from a single copy of the region that you want to amplify from, and then you add your primers, and then now you have 2 copies of that region. So you have that original piece of DNA that's very long, and then you have a piece of DNA that starts where the primer is and then goes past, and then you have the opposite that starts from here and then goes past. And so this is an exponential process. You do multiple rounds of this, and so each round you're doubling the amount of DNA of that particular region. So initially you had 2 strands that could serve as a template, and now you have 4 strands. And so all 4 of those strands bind primers. So primers bind to all 4 of those strands, and then as a result you have 8. And then you do this over and over. It doubles each time, 2, 4, 8, 16, 32, 64. And if you do this for about 30 to 35 rounds, you end up having a ton of just this region of DNA. So we've amplified a small region of the genome that we're interested in, starting from a DNA template sample. And so in order to successfully amplify a piece of DNA, you have multiple temperatures that you cycle through. So they're called the DNA denaturation temperature, they're called the primer annealing temperature, and they're called the primer extension temperature. And so the DNA denaturation, this is like 99 degrees Celsius, so this is almost like boiling water. And so the DNA becomes so hot that these strands end up separating from each other. And then you have the primer annealing temperature, and this is a temperature where the primers are able to form this double-stranded bonds through hydrogen bonds with the template DNA. So these two original strands don't actually anneal together. It's this primer that anneals to this template strand, and then on this opposite one that this primer does. And then you have the extension temperature, and this is where the polymerase is active and it extends from the primers in order to create these new strands. So you do these rounds of 3 temperatures over and over and over again, and then as a result you have, a ton of DNA. And so in order to successfully do this, what you need are primers that are very successful at annealing at this temperature. So typically you use something like 55 degrees Celsius, and so at that temperature you want the thermodynamics to be such that these primers will anneal just to this region. And so if the melting temperature of this is too high, if this primer is too long, then what happens is it'll start to bind non-specifically to other regions of the genome, because there's enough energy at 55 degrees that it can bind to these other regions. But if the melting temperature is the opposite, then what happens is it's not able to actually form these dimers at this temperature because the thermodynamic energy is higher than the energy that holds the hydrogen bonds that hold these together. And so that's what this program Primer3 does for you, is it designs primers that can be used to identify good primers for amplifying a region of choice. And so if you're familiar with DNA polymerase, which I expect many of you are, if you're familiar with PCR, then this is probably pretty straightforward to you. If you're not, then I encourage you to kind of go over this in more detail, or maybe consult Wikipedia, or there's all sorts of tutorials on how PCR works, or talk to me and Tucker, during class. Part of the class time is to make sure that you understand the process of PCR, so you understand kind of the point of this program and kind of the point of the assignment that we're going to do. So why does the sequence of the primer make a difference to how effective the primers are going to be? And so that gets into the biochemistry of the hydrogen bonds that form between the nucleotides. So remember that thymine makes a hydrogen bond with adenine, and cytosine makes a hydrogen bond with guanine. So the complementary nucleotides are T and A, and then C and G. But the number of hydrogen bonds that they form is different. So there's only 2 hydrogen bonds that form between A and T, and there's 3 hydrogen bonds that form between C and G. So if you had primers that were only made out of A's and T's, then the melting temperature is much lower because there's not as much energy to hold those things together. Whereas for C's and G's, because there's 3 hydrogen bonds per base pair, it takes a lot higher temperature in order to separate those together. And so this graph just kind of shows you, the melting temperature, the amount of DNA that is hybrid or that has melted and is a monomer versus those that are in a hybrid duplex. If you had a poly AT, a poly GC, or just naturally occurring. But the idea behind this is poly GC is much more effective in holding DNA together. And so when you design primers, what you're trying to do is design primers that anneal at this primer annealing temperature right here. And that's typically between 50 and 55 degrees. So Primer3 is a program that does that for you. So one of the things it pays attention to, there's a website version of this. So one of the things that it pays attention to is the melting temperature. And so, you can choose a melting temperature that you're trying to target. But it also pays attention, so if you go to general settings, like this is one of the things that you can specify, is the primer melting temperature. So you can say the optimum value is 60, but anything between 57 and 83 is acceptable. You can also specify things like the primer size. So if you want to have an optimal primer size of about 20 base pairs, and then you can have a range between 18 and 27. So why might you do that? The more expensive, the longer the base pairs, the longer the oligo is, the more expensive it is to produce it. So you might want to try to design something that's shorter for that reason. You can also specify things like GC content, and that also goes into the choice of primers. So it might try to optimize at 50% GC. So 50% of the nucleotides are Gs or Cs, and 50% are As or Ts. You can specify the product size range, so how long you want your piece of PCR to be. So if you're trying to do 500 base pairs versus 1,000 base pairs, like all of these things are actual things that you can specify. And then it also has these other things that it looks for, and in general you don't really need to know the details of these. I typically never would modify any of these values. But the idea behind this is that there's other things that can happen that can disrupt your ability to do PCR. So if the primers, for example, are complementary to each other, then those primers might actually for primer-dimer pairs, and instead of annealing to the template, they anneal to each other, and then you never get your PCR to work. Or they can anneal to themselves and form these like loops. So these are all things that Primer3 actually looks for. And typically, like the research group that thinks a lot about PCR, that spends their life dedicated towards researching it and reading all the papers to it, they're going to have a lot more knowledge about this stuff than you, and so that's why you kind of want to use one of these programs. So you could spend your time designing your own primer choosing program, or you can spend your time learning how to use a software that was developed by someone else. And typically it's almost always better to learn how to use the software that exists, because they'll have put a lot more thought into things that you don't have the expertise, or you would have to spend a lot of time to gain the expertise in order to design yourself. So for that reason you want to use some sort of program. And so Primer3 is just one example that you could use. And so if you want to use it, you can paste a DNA sequence that you want. You can give it a name, so we can call it test region, and we can specify, for example, we want something that's 500 to 600 base pair long. And so we run this, and now it has picked two primers that will give me something of the appropriate size. So it starts at the 107th nucleotide. The length is 20 base pairs. The melting temperature is 60 degrees Celsius. The GC content is 50 degrees. And it also reports kind of these other as well. And so the second primer is here, the yellow. And so because this is the reverse complement, you'll notice that the primer sequence is actually the reverse complement of the sequence that is shown for this. This is the sequence right here. So this AAAA, that is the sequence of the primer, and then you'll notice this ATCT, that is complementary of this. So ATCT is the reverse complement of this region. So it's useful to use this this web WebBrowser in order to learn how Primer3 kind of works. It's a lot easier to learn something in this graphical manner, and then eventually you're going to come back to the command line in order to use it. So make sure you understand that the sequence of this primer and why the sequence of this primer is the reverse complement of this region, while the sequence of this primer is just the exact sequence right here. You'll also notice that it returns multiple primers. So if you want to design multiple primer pairs to amplify the region, it gives you different possible products, and you notice that, for example, the product size is different. So this is a 540 base pair, 585, 600, etc. So they all these primers are distinct from each other, and all of these are typically pretty good. So you can also exclude certain regions. So if you don't want primers picked from a certain region, you can do that. And you can either do that by modifying your DNA sequence so that you add these less than or greater than brackets. And so, whoops, not save sequence. So, by doing this, now we exclude choosing the primers from this region. So let's, do this region right here as well. So let's go back, and we are going to do this. And so now we've prevented the program from picking primers in this region. So you'll notice that the primers that are reported are now different because of that. So the excluded regions just says, don't choose anything from this region right here. And you can kind of do that wherever you want. So you notice now it picked a primer here and a primer here. So the primers cannot be chosen from this region. And we can do this either by using these brackets that we put into the sequence, or you could specify by using this syntax where the first is the position and then the second is the number of nucleotides to exclude. So starting from position 100, and then the next 200 bases are excluded. So there's basically two ways we can use this excluded region that way. So you can also target, for example, and so this means that you want to make sure that this region is amplified. So you might do that, for example, if this is like the repetitive region and you want to make sure you design primers that get the entire repetitive region. So this makes sure that one of the primers is selected on the left-hand side and then one of the primers is on the right-hand side. And so again, we get multiple options for primer pairs that would work for that. And so again, you could also use that syntax. So this has actually just like translated what my brackets were, but you can also use that syntax in the targets. So there's kind of two different options that you can use. And then finally, included region is probably what you think. And unfortunately, the issue, let's see if I can find one. Included region forces it to choose it between the region that you're saying. So the reason we got an error before was because the included region was smaller than the product size, and so it wasn't able to find any primers that were within the included region that were the appropriate length. So play around with this and make sure you understand how you can use the web template in order to pick PCR products that you want to choose. And next we will get into the command line version of this program.